I don't know. No idea.
Keep going. We're still in chapter 7, genetics. We're just quickly going to do horizontal gene transfer mechanisms. This thing, your handouts package. All right, third test. Everything's on it. Mostly viruses and metabolism and that stuff. But, okay, never forget your trees of life. Then you have definitions. Here's control of microbial growth, right? Sanitization, de-germing, disinfectant, blah, blah, blah. You have oxygen requirements, pH requirements, temperature requirements.
Where? Which page?
I'm going through page by page. Then you have host-microbe relationships, just more vocabulary. Epidemiology, more vocabulary. We're doing chapter 14 soon, not yet. Endotoxins and exotoxins. Here is your cellular respiration summaries. Anaerobic respiration, take your glucose, split it into 2 pyruvates, end up with alcohol or lactic acid. You get 2 ATP. Aerobic respiration, you need oxygen. Oxygen's required at the Krebs cycle step. Oxygen plus glucose, your first step is still glycolysis. Glucose split to 2 pyruvates, and you get 2 ATP. But with oxygen that enters at the Krebs cycle, you get 36 more ATP because you break your pyruvate all the way down into carbon dioxide and water and heat energy. Don't like it that way? Maybe you like it this way. I actually stole this from one of those Cliff Notes books and I modified it a little bit. And mine has a little red ink or blood on it. I don't know what that is. Glucose at the top. Split it into pyruvic acid and glycolysis. That's your first step. You get 2 ATP. If you put the electrons and hydrogens onto the pyruvate, that's fermentation. You get alcohol or lactic acid. Let's say you decide to go the other way, aerobically. So if you go the other direction, you get your 2 NADH, your electrons and hydrogens transferred, and your pyruvic acid enters the Krebs cycle. Here's the transition step. Has to be made into acetyl. CoA enzyme, CoA picks it up, takes it into the Krebs cycle. You're going to do 2 turns of the Krebs cycle per molecule of pyruvic acid. You get electrons and hydrogens, go through the electron transport chain, resulting in more ATP. So if you add them all up, you get 2 ATP from glycolysis and fermentation. In anaerobic and aerobic respiration, you get 2 ATP from glycolysis plus 36 more because you did Krebs cycle and electron transport. That's what that is. And what's their end product? Water. Oxygen gets reduced to water because oxygen receives all the electrons and hydrogens at the end. In fermentation, it's pyruvic acid or pyruvate that receives electrons and hydrogens, and it gets fermented into alcohol or lactic acid. Okay, next one. Bacterial gene transfer mechanisms. That's what we're going to talk about right now. Okay? So keep that page in mind. Next, genetics. We did this yesterday and the day before. Definitions, types of mutations. Mutagens. Then you have a strain concept. We've already talked about strain. Genetic variation of a species, not a strand. And then this other stuff is just a quick summary of the immune system. Active immunization, passive immunization. You'll need to know that. Don't worry about the blood typing. I just used it as an example. Then I put a blood typing chart, makes blood typing easy, if you remember that from anatomy and physiology. And I just put some isolation signs, one isolation sign, just for, information. And then different types of microorganisms that we studied. You've got your eukaryotes, you've got your prokaryotes, and then you've got lots of viruses. You have RNA viruses, which can be positive sense or negative sense, and then you got your DNA viruses, which can be single-stranded or double-stranded. So I just wanted to walk you through your handouts package so you know what's in there. Because it's kind of a short summary of a lot of the stuff we covered. Okay, bacterial gene transfer mechanisms. That's what we're going to do quickly because we're running out of time. And we want to have one more test before the final. So, I scheduled your third exam for this weekend. We might make it, we might not. I'll let you know tomorrow. But if I were you, I wouldn't take it tonight, or Wednesday night. I wouldn't take it until I knew we covered everything, okay? So don't be silly and take it before you're ready, because we're not quite ready. I'm sorry, my computer's not quite ready either. Taking its time.
Are you going to review for the third exam?
Absolutely. That's why I say I wouldn't take it before the review. I want to look over it myself and see if I make sure I covered everything that's on it. Alrighty. Okay. Remember the central dogma of how genes code for proteins. This first part is DNA replication. The DNA is being replicated, see? Then you have DNA. DNA is transcribed into RNA. RNA is translated into protein. DNA to RNA to protein. That's the central dogma, they call it. Then you have all these other PowerPoints that kind of go with the lecture I gave you. Picture of DNA molecule, comparing bacterial genome. The book talks about sizes of genomes and stuff like that. I left a lot more PowerPoints than you might find helpful, different mutations. Okay, we want to do a horizontal gene transfer. You may remember these terms, as I mentioned them before. Okay, what do you need to remember? When bacterial cells replicate by transverse binary fission, or your textbook author just calls it binary fission, one parent cell copies its nucleoid, splits in half down the middle by building that transverse plate or septum, and ends up with two daughter cells with identical copies of the DNA. Each daughter cell has one strand that's original from the parent, and the other strand that's the copied strand. That's semi-conservative DNA replication, okay? If a mutation happens, then the copied strand might be a little different. So when that daughter cell goes to divide and her strands are replicated, one of the strands that's different is going to result in a different daughter cell. So, mutation is one thing that can give new genetic types to bacteria. Mutations during transverse binary fission are rare because when the cell copies its DNA, there are proofreading enzymes. The proofreading enzymes are going to check for mistakes. I happen to be a professional proofreader, and I usually catch them all, but you know what? There's always one that slips by you. So the rate of mutation is rare. It does happen even with the proofreading enzymes, but the rate of mutation is slow. Mutations are rare. So when a cell divides by binary fission, it results in daughter cells that are clones. They're genetically identical, unless a mutation happens, and that rarely happens. The other way bacterial cells can get new gene types is by horizontal gene transfer mechanisms. Horizontal gene transfer mechanisms is another way bacteria can get new genetic types. So that's what we're going to talk about now. Transformation, transduction, and